So for those of you that may be patients or become patients and are going to go on immunotherapy, no matter which physician in town or which town, we would very much like to work with you to get a sample of your stool so that we can sequence it and figure out whether you have the right bacteria to respond to the immunotherapy. And if you do not, we are developing probiotics which could enhance your chance of responding. Okay? So that's the theme of the talk. Now I'm going to take you back. The ERK and Earth, let's go back to that. So 4.65 billion years ago, which was at the beginning of our solar system, a mass the size of Mars came and banged into the Earth and ejected the moon. So I'm showing you a picture of the moon. After that, around 4.5 billion years ago, the heat from the iron at the core of the Earth, because of gravitational condensation, there was so much heat that it liquefied the center of the Earth. And periodically, the magma in the center of the Earth from the gravitational condensation, the radioactivity of uranium, strontium, and potassium created so much heat that it would bust through in volcanoes on the surface of the Earth and emitting both the magma, which you're familiar with, but also emitting hydrogen gas, nitrogen gas, CO2, the early atmosphere, and water vapor, the early atmosphere of the Earth. This was obviously not a great environment for the formation of life on Earth. And in fact, the sun at that time was a blue-white star, meaning that its temperature was over 7,000 degrees and it emitted a large amount of ultraviolet C light, which would have burned us to a crisp if we were around at that time. But things cooled off. A big event occurred 3.9 billion years ago. And that was called the Late Heavy Bombardment, or the lunar cataclysm. You see these pop marks on the moon. They represent an event that occurred between Jupiter and Saturn. They came out of orbit a little bit in the early formation, in the early days of the solar system, and that caused a large piece of the asteroid belt to come flying down, landing on Mars, the Earth, Venus, Mercury, and the moon. Okay. What's important is those meteorites brought the key ingredient for the formation of life on Earth. Okay. So there were two major types. I'm going to show you, and I can pass them around for you all to look at. This is called dendritic meteorite. It's very large amounts of carbon. You can pass it around. And as it came down and splashed into the atmosphere in heat, it created something called hydrogen cyanide gas, not something you'd like to run around with, but it's a very effective chemical ingredient for the formation of other compounds. The other key ingredient that led to the eventual life on Earth is called shrivosite. I'm going to pass around this meteorite material. You see it's very tiny. I want to get a much bigger one, but my wife won't let me spend the large amount of money it would take to get a big sample. So you're going to have to look at this. It's composed of nickel and iron phosphide. And the key ingredient there that led to life on Earth was phosphorus. The early Earth did not have much phosphorus available to it, certainly not on the surface. And this then allowed life to form within 100 million years, and I'll tell you the steps to that. This just shows some of the meteoric showers. What did I do with my... I lost my little clicker somewhere. Here it is. Okay. This just shows what happened on the moon. The Earth had 1,000 to 5,000 times as many meteorites hit it. So it was really a big event. When that occurred, the materials that I described that came down reacted with the bedrock metal oxides, hydroxides, chlorides, carbonates to create new compounds. And this provided feedstocks and that as water came down from these volcanic mountaintops and worked the way down, then you had congruent synthesis or convergent synthesis of new compounds, including the formation of sugars, amino acids, nucleotides, because of the ferrocyanate chlorides and phosphates that the meteorites had brought. And finally, here's another one that I'll pass around, another. This is olivine. This is a phosphate, so it's a late product, very beautiful. It used to be blue-green, but the iron in it, when it's exposed to light, loses that and it looks black. And that produces phosphate, which you need for your nucleotides to make your nucleic acids, to make us. And then finally, this morlite clay is an aluminum silicate that provided the basis for the phosphate compounds to line up and make polynucleotides, which led to the formation of life, as we'll see coming up next. So this all occurred 3.8 billion years ago. And the first thing that happened in the ocean beds that were starting to form is something called liquid phase condensates. And this is where the polymers of sugar and the polynucleotides could combine together, separate themselves from the rest of the water environment to make something called a protocell. It's not yet a cell, but it's on its way to make a cell. And this occurred very rapidly. You know, I was going to bring you something to demonstrate it, but it looks a little goopy or silly, and that is Jell-O. Anyone those are old enough like me to have eaten Jell-O when it used to be popular. If you put Jell-O in a glass of water, it doesn't automatically dissolve right away. And that's a protocell because it forms its own sort of separate constituency. Now, actually, ice in a glass is a protocell, although obviously it will melt as the temperature increases. Now, what I'd like to do, and I'm not sure I can do it on this computer, is show you what happened in early life. The RNA formed a, let's see where formed a ribozyme. Let's see if I can get this to work. Whoop. Oh, shoot. Well, I'll have to tell you. All right. The RNA polymer formed a ribozyme that could then copy itself and make duplicates. And then it had one other property. It was able to make other compounds if it had an energy source, and you can't see it because I can't get this to work, but it will divide on its own. So any of y'all remember Alan Turing? I think they had a movie about him. He was the one that started the computers, and he also solved Enigma in World War II. Well, among other things, he also solved how early life could divide and make offspring if you provide energy. So at 3.8 billion years ago, in the bottom of the ocean, there are these hydrothermal vents where hot water comes up. And if the hot water is exposed to olivine, this is a mineral, you see it has two different types of compounds in it, ferrolite and phosphorite, and they react with the hot water to produce the key material, the energy source to allow life, hydrogen gas. Okay. And when it does that, it produces hydrogen gas, and the other products of that are silica, magnetite, and serpentinite. So I'll pass it around. So these are fossils. Oddly enough, they're rocks, but they're fossils that show you the beginning of early life before we even knew about it. Okay. And they can be found at these hydrothermal vents. And you say, well, I don't know much about hydrogen. Well, hydrogen gas was used in some of our early rockets for the United States space program. And there are even people that wanted to try hydrogen-powered cars because they'd emit water, you know, as a product, but that hasn't really taken off. We're using battery-powered cars, I guess. 3.4 billion years ago, there's evidence of sulfobacterium in Australia found and deep embedded in the rocks. So life formed a long time ago. The reason I bring this up is early life was bacterial. This organism was called LUCA, last universal common ancestor, but we know it today as archaeobacteria, and you know it in each of your bodies is Methanobrevibacter smithii. You don't talk to it, but it's in you. It's the relic, the like living fossil of that original organism. And what it does is use something called the Wood-Lunchthal reaction to take hydrogen gas that's in our gut and convert it into methane. So the principle of my first part of my story is that every time you pass gas, you're connected to the man in the moon. Now, I know this is weird, but I love that story, and I use that as an excuse to my wife. All right. Right. Now, the reason that's important is that the bacteria in our bodies have been around a long time, and they really have adapted to many different environments, but they also help us to be alive. So this is that early beginning, but you can see for a long period of time, there were no more complex organisms. For over 1 billion years, bacteria and archaeobacteria sort of grew, and they were in the ocean, they were around. It's just bacteria. There were no humans, there were no bigger animals. Then something interesting happened. There were something called cyanobacteria formed, and that's a type of bacteria that used manganese for its energy. It was able to do a redox reaction with the metal manganese, and that allowed a growth of the cyanobacteria. And then the biggest event occurred 2.35 billion years ago, and that's when cyanobacterium discovered a magical way to do a redox reaction that was very powerful. They took sunlight and broke water to produce oxygen gas, and that's photosynthesis. So that happened. It's just one of these bacteria a long time ago, but it had a big event. The CO2 in the atmosphere fell like crazy. The oxygen went way up, and it was the opposite of what's happening now with global warming. And in fact, we had a snowball Earth. This actually happened more than once, but I'm going to talk about the main one. And that happened 2.2 billion years ago, wiped out a lot of the organisms. Luckily, water goes underneath the ice, so there were still living organisms underneath the ice. Then something called the Loma-Jundi Jatali excursion occurred. So this is named after two scientists who discovered there was an event where the oxygen got so high that it was like using Clorox on life. It was killing off everything, but a special organism found that if it used cytochromes with copper, it could do oxidation reactions to then stay alive, but only if it hid within another cell. And so within a bacterial cell, squeezed inside of an archaeobacteria to produce Loki, the very first eukaryotic cell, and that occurred 2.15 billion years ago. And then after that, there was a rapid evolution. The evolution occurred because we're used to the North Pole and the South Pole, but for 800 million years, the poles kept swinging around. So the North Pole might be in the Arctic one year, and then 50 years later, it's over in South Carolina. You say that can't happen, but it did. And that went on for 800 million years, called rapid evolution of organisms. Okay. If you look here, eukarya is what we come out of. There are 4 types. They're protists. They're just these little paramecia, single-celled organism. Unless you happen to be unlucky and swim in a pond in Florida with Naegleria and get killed by it, you probably aren't going to, or if you go to Guatemala and get amoeba or amoebic dysentery, you're probably not going to have a lot of knowledge or interaction with protists, unless you're a high school biology teacher and you have a little paramecia. But in addition, there's fungi, and we'll talk about fungi a little later, but fungi both have mycelia and they also have the mushroom phase that you're familiar with, and there are other fungi as well, and then plants and animals. So there's very few phyla among the eukaryotes. In contrast, there's 92 bacteria phyla and over 30 Archaeobacteria phyla. So higher animals are really a tiny part of life on Earth. Okay? That's really the theme that I wanted to cover, and that they've been around a long time. We're sort of newcomers. Okay. So what does this mean? So now I'm going to talk about how these bacteria relate to animals such as humans. You notice in the phyla, there's a lot more phyla in the soil and the oceans. As you get inside the fish gut and then the mouse gut and the human, there's fewer and fewer phyla. And a limited number of species, although more of those, there's maybe 50 to 70 genuses and 300 species. And what this resembles is something called adaptive radiation. And this is what these bacteria are making a relationship with the animals. And I wanted to start with this because this is work done, as you know, in the 1830s to 1860s, first in the Galapagos Islands, where they found that a single species of finch had gone and evolved separately on the different islands, but they're all related. And that's what's happening in your gut. You have just a few phyla, that Methanobrevibacter smithii from the Archaeobacterium, and a few other bacterial phyla, and then they radiate out, making a larger number of species. And this came out of Darwin's book describing it. And this is Jay Gould, who helped him with the birds, and then there's Charles Darwin. So it was true not only in the Galapagos Islands, but the discoveries were made concurrently in the Tahitian islands with snails. Okay. And that's what's going on, the evolution in our guts. Okay. But what does that mean for us? Well, we know that the bacteria in our gut helps us make vitamin K. We need that for clotting. It helps us degrade, stuff we eat so that we can get nutrients out of it. And finally, it detoxifies a number of poisons that may come into our body. But for fun, I wanted to show you that it's not unique to humans, this relationship between the microbes and the either plants or animals. So these are rhizobia. They're the roots of plants. They consist of bacteria that fix nitrogen from the air and convert it into compounds that a plant can use to grow. And without that, they'd be in big trouble. Now, that's not as true now, thanks to the very evil scientist Haber in the 1918, 1915 era. He discovered how you can make ammonium nitrate, which has revolutionized agriculture. So you don't need this as much, but he also made all the poison gases in World War I and some in World War II. So I hate him. He's gone. But plants do it on their own with the help of bacteria. Okay, termites. So you say, well, we don't have that many termites, and we pay for an inspector. Termites eat wood, but they actually don't have the ability to digest the cellulose. They use bacteria to digest it in their gut. Not only termites, but it used to be, and we still have some places where cows will eat grass. Well, grass is a cellulose type compound. If we tried to eat it, we wouldn't digest it. Cow can't either, but the bacteria in its rumen can. Okay. Aphids, if you're into gardening, these are these tiny little insects that suck the nectar out of the phloem out of the plant, but they cannot make amino acids. They have to have bacteria in their gut to make the amino acids in order to live. And this is a squid. Squid live deep in the ocean, and you say, well, why do they need bioluminescence? This bioluminescence. Well, one, it helps them find each other, but also, oddly enough, there are predators that look down, and if they see a dark shadow where there should be light coming back, then they know they've got a target. So they use this for defense, and this is the bacteria from their light organs that produce that, and that's how they get the bioluminescence. They don't have any intrinsic ability to do that. Then you're already familiar with coral. Coral's color is due to photosynthetic cyanobacteria. When we bleach the coral because it's either too hot or too acid, it's going to kill off the coral because it needs those colored cyanobacteria to provide it the food to survive. So those gives you an evidence that there's a serious relationship between the microbiome and the animals in which they reside. So now we're getting to humans. How does this work? So I drew the bacteria as like little rods and some food fibers, hopefully not chicken salad. And this is your epithelium in the colon. You see a mucin layer that's produced by goblet cells. Paneth cells produce antimicrobial peptides to help reduce the number of bacteria that can get through. But oddly enough, your gut colon has M cells, which are very thin cells that allow bacteria to go across. But those same M cells produce a chemokine called CCL20, which attracts dendritic cells, all right, and actually helps create this lamina propria area. And the bacteria then are swallowed up by the dendritic cells, carried to the mesenteric lymph nodes, where they educate T and B cells to counteract the bacteria, so that we have a homeostasis. So we have 100 billion bacteria in our colon, which is equal to the number of cells in our body, and yet we're not being killed by them. And it's because we've come to peace. We have sort of an even fight between the two. Some of these cells go back, B cells, plasma cells, to make IgA, which counteracts some of the bacteria. And as well, there's T cells and T regulatory cells to keep peace in this area. Okay? So what happens if you are born without any bacteria in your gut and you live in a bubble? They tested that in animals and rarely in humans, and it really messes up the immune system. So our immune system is partly educated by the bacteria, so it works both ways. We need the gut bacteria for our immune system, for our behavior, for our metabolism, and we'll show you evidence for that. So the best way to find evidence for any biological process in humans, not in the laboratory, but in humans, is to see where there's pathology and see if you can trace it back to what's going on. And there's evidence of an overgrowth of one bacteria, Clostridia difficile, that you have a dysbiosis in your colon. The other bacteria they've been beaten down, as you'll see in a second. Graft versus host disease, which can affect multiple organs in the body after an allogeneic transplant, is affected by the gut bacteria. Inflammatory bowel disease, no surprise that if you have the wrong bacteria in your gut, you can get inflammation. You can get irritable bowel, you can get Crohn's disease, you can get ulcerative colitis. This is sad. Obesity and metabolic syndrome are at least in part affected by the gut bacteria. And whenever I gain weight, I don't take the blame. I'm blaming it on my gut bacteria. And I try to change them, and it's not very effective, as I'll show in a second. Interestingly, autism has been shown to have a strong component related not to vaccines, of which there's really no evidence, but a whole lot of data suggesting the gut microbiome does influence behavior, and if you can get the right gut microbiome, you calm down the autism. Not completely cured, but you can dramatically improve it. And then finally, my main talk will be on cancer therapy. But I want to give you some of these to give you a context, since most of this audience, I see there's some superb scientists in here, but most of y'all don't do science every day. So we're going to have a little fun to show some evidence for the importance of the gut microbiome. So here's your colon if you've got too much Clostridium difficile. You can see ulcerations. This is serum that's coagulated in the gut, and this is with a little scope, colonoscope looking up in it. You see blood in it. And the treatment for this, the best treatment is not antibiotics, which has a cure rate of 30% with vancomycin, which we used up until 3 or 4 years ago. But in fact, donor feces, if you give an infusion of that, your cure rate's 94%. And you say, well, maybe that was discovered 4 or 5 years ago in the Netherlands. We probably aren't doing that here in Mobile. Not true. De Palma does it routinely here in Mobile on patients because you don't even require FDA approval for this. and you can even do it at home if you need to. But they're able to do it. And what that does is it takes these bad Clostridium difficile, and if you look at the stool by sequencing, and this is just 16S ribosomal sequencing, you see that after a donor transplant, this is day 0, by day 14 it's been reduced dramatically, sort of wavers a little, but stays under control. At this low level, the patient is not having overt disease. In fact, Clostridium difficile is in all of us to some small degree.
Now, how do you do that? You're actually saying you take somebody's feces and
Correct.
put some in your butt?
Correct. So the way they do that is you can you take some feces. You want to take it from as normal a person as you can. So in the early studies they did in the Netherlands, they had one of the donors was overweight, and they filtered the stool. They gave it either by rectal suppository or by a nasogastric, nasojejunal tube, so it would go past the stomach. And they infused that, and the very high cure rate, but the one that got the obese patient stool gained 25 pounds. So there is so they have to be careful now when they select donors. They're careful obviously about things like hepatitis things, but also other properties. And you really are not going after someone with Asperger's syndrome or obesity or some other problem.
Have they done anything locally on autism?
I wanted to. So I have begged USA, so if you can help me, they're not doing that type research. They are at UT Southwestern where I came from. They even had a grant. They're not doing it here, but I would love to do it with people here. They have a little bit bigger program here with PTSD that they were interested in looking at, but you tend in science where there's you look where, like you look under the light bulb. I mean, there's obviously truth everywhere, but you don't look in the darkness. So you go where there's already a hint. So since there's pretty good data on autism, rather than looking at depression or PTSD, you sort of want to concentrate on that. There's even good animal models. So no, I'd like to do that because it's a serious problem. I've had some of my best young faculty where they've had a child with autism, and then they had to leave. They had to go to a place where they had a good autism clinic, for their child, and it can be very tough. All right, well, let's go through these things. So graft versus host disease is where your own T cells are attacking yourself. Remember that. But they're not really yours. They came from the donor, from the transplant, right? And they're trying to kill off the leukemia or lymphoma, but they can attack the skin, the lungs, the liver, the gut. If you have Blaudia in your gut, then you don't get the graft versus host mortality. Look at the difference. All right. So they're now considering doing studies giving Blaudia to people before transplants up at Sloan Kettering. Here, for lung disease, if we look at the incidence of pulmonary complications, if you have proteobacteria, then the pulmonary complication is much higher with the proteobacteria. In its absence, you don't have it. So it varies for the different organisms. You can see that the gut is communicating with the immune system. Now, what about inflammatory bowel disease? Really is a form of inflammation or immune system dysfunction. And this is an electron micrograph of the surface with these damaged mucosal cells. And fecal transplants have been used. Now, there's still ongoing studies. The numbers may not end up quite as good. Crohn's disease, there are several publications suggesting a 70% remission rate. Ulcerative colitis, around a 40%. The reality is always the early reports are better than later reports. So if you cut that in half, it's probably what it'll be. But that's still impressive because these are horrible diseases that usually require removal of the colon. So let's say that's 40% or 35%. For those people, that's going to be great. And fecal transplants can work, and they're not expensive. All right. Now, I used to have up in this area a picture that had the incidence in the different states in the United States, and I've been giving this talk, obviously in the South. And I saw that the audience didn't really like that picture, so I won't show you that. But I will tell you Alabama is up there. It's like number 2 or 3 in obesity. But I'm from Texas, and it's not a whole lot better. I blame it on our Tex-Mex food. But the South in general is in very bad shape. So unless you plan to move to Colorado, this is interesting information. And here, transplants have been used. They have not cured obesity, but they have improved insulin sensitivity, which is an early marker of the metabolic syndrome. Probably going to take more work to make much more headway. And we'll explain why that is. Now here's your autism. So autism is a multi-symptom spectrum disorder. At a physiological level, it's really ugly. There are actually changes in the synaptic boutons in the nervous system. So we're not just going to come in with something and make it all go away. But the animal models show that depending on how the mother is treated in terms of her gut bacteria around the time of pregnancy and then early delivery, that has produced autism-like syndromes in mice where they do repetitive behaviors or they avoid other mice. And in fact, if you look for transplant, and this is a very nice transplant regimen that was done, and they actually used fairly mild child-like bacteria, Bifidobacterium, Provitella, the Sulfovibrio, which are not uncommon in children, that the bad activities go down versus control, and the good activities improve. So it's not absolute, but it's a hint that this is an active area of science, and we actually know there are some metabolic products from gut bacteria that can influence the brain and how it acts on some of these functions, different for different ones. Yes?
So are you saying that it's the bacteria of the mother when she's pregnant that is passed on to the child?
In the mice. We don't know about humans. I was telling you the mouse model. What we do know in humans is there's dysbiosis in autistic children. Many of them have bowel dysfunction as well. So, but I want to be careful. You don't want to overstate the associations. I'm really using this part of the talk to just show you that the gut microbiome influences a number of different things in our body. And in fact, if you look on Amazon, you'll find books on the gut brain, as well as allergies, inflammatory bowel, other things. A number of good books. And in fact, I brought one for those that want to have fun after this talk. This is a lay book by Ed Young called I Contain Multitudes. And if you get through that and you like it, then I've got more advanced books to get you further into it. But that's a fun one.
One other question.
Yes.
Cesarean versus vaginal birth. Do you see a difference in the
Very,
microbiome?
very severe. So as a result, in the United States, not in some Scandinavian countries, when they deliver the child, someone takes their hand, goes into the vagina of the mother, swabs out material, and then coats the baby with it. Okay, that's standard practice now in the United States. Okay, so chemotherapy actually, the response to chemotherapy is also affected by bacteria in complex ways. And if you have a lot of Fusobacterium nucleatum, then that's not good for a response to FOLFOX chemotherapy. If you have gram-positive bacteria, oddly enough, and CLL and lymphoma, you do a little better with chemotherapy. If you have lung cancer, non-small cell lung cancer, and you have Enterococci hirae, that's a little better. You notice these things aren't cure, all or nothing. I wish I could tell you that. I spend all my time, there are scientists here that get beautiful results. I always get results that are 10 to 20%, so it's like painful. I have to repeat the experiment 3 times to believe it. But there is an effect. And same in acute myeloid leukemia, Eubacterium lamosum is better. You have longer survival. So there's already, without immunotherapy, a hint of the value of the gut bacteria. You say, well, how is it working there? Well, it turns out probably our response to chemotherapy is mediated by the immune system, not by just killing tumor cells. So that new thought for how chemotherapy works is sort of slowly taking over the oncology field. All right, So now we're going to switch gears. I won't tell you any more about other side things. I will tell you an interesting thing. Arterial sclerosis is increased with certain gut bacteria. It's not on my slides, but they found the compound trimethylamine oxide that comes out of the bacteria in the gut, goes to the bloodstream, gets to the blood vessels, and damages the coronary arteries. So there's work in that area as well, but it's not as far along, maybe in a future talk. But what about cancer immunotherapy? What are we dealing with? So these brown color is immunohistochemistry. I've got someone who's been trying to help me get it work here, but this was done in Dallas, and this shows a tumor loaded with T cells. If the tumor is loaded with T cells, we're going to see in a second, it's more likely to be able to respond to immunotherapy, particularly if it's a T cell activating immunotherapy. Sort of common sense, right? Now here you have a tumor with very few cytotoxic T lymphocytes. And guess what? That tumor is less likely to respond to immune checkpoint therapy. So what can we do about it?
You test for that first before you would treat them with immunotherapy?
Well, that's a good question. You know, immunotherapy does have side effects. And people are talking about putting together a algorithm for what things would predict response and what wouldn't. At this point, that would certainly be in the algorithm from most laboratories, but we don't do that right now. And the reason is we have so few options for people with metastatic cancer that even if the response rate goes down to 10%, you probably are still going to use it. But it's still useful to have that knowledge, and we're trying to get better at it. Why? Because immunotherapy does have side effects, as we'll see in a second, so you'd like to pick the right patient. Or find a way of getting more T cells to go to the tumor before you treat. And that's really my research. All right. So how does immunotherapy work? So here's the tumor cell. There's the T cell, brown in color over here, but blue up there. And it has little proteins sticking off the surface. And interestingly, it has proteins that tell it to stop attacking. And so why would you have that? Because you've got a lot of different antigens in your body, and if T cells just went willy-nilly and attacked you all the time, you'd have lupus all the time, you'd have inflammatory bowel disease all the time, you'd have encephalitis, your immune system would kill you. So we have what's called immune checkpoints that block the activation of the T cells, and our normal cells have the ability to signal that. So PD-1, PD-L1, B7, or CD80, CTLA4, tells the T cell, no, I'm normal, leave me alone. The trouble is, guess who figured out how to use that mask? Cancer. So almost all cancers do that. Some don't need to do it if there's no T cells there, but it's a very common mechanism so that a tumor can still grow even when it has T cells in it. And it was really the discovery of James Allison and then others that led to blocking that, activating the immune system, which is good and bad. So here's as of 2 weeks ago, and I can say now because, you know, I have on my cell phone notification if a new drug is approved in immunotherapy. But as of 2 weeks ago, these are all the approved immune checkpoint antibodies, the different cancers, and the response rates. Now, if you notice, it isn't bad if you happen to have Hodgkin's lymphoma that's relapsed or Merkel cell cancer. Now, those don't relapse that often, but there you do pretty well. Even melanoma is not bad, although we'll see in a second it comes with serious side effects. But as you get down to the others, you're 50% down to 12%. So we got to do better if we're going to really help people. The other problem is whenever you turn on the T cells, you can start attacking your own body. So I'm embarrassed to say it may not be true today, but my fellows Ashish and Pranitha have had the pleasure or lack of pleasure of having my patients come into the hospital on a regular basis with autoimmune complications. And you get all of them. I mean, there's some that aren't even listed here that I've seen, orchitis, but some of these like uveitis in the eye, It says 1%, but you see more than that. Hypophysitis, thyroid. So all these are different organs where the T cells attack you. A good reason for either not treating someone if they're not going to respond or trying to figure a better way to make the response rate higher. It's a bad, sad situation if someone doesn't respond, but they get a serious complication. So what can we do about it? When I was in Dallas, a group in Chicago had done a study where they were looking at mice and they were using immunotherapy, these immune checkpoint antibodies, and they had this melanoma in mice. And when they gave the immunotherapy, the tumors went away. And they were repeating the experiment, but it was wintertime, so they had to get the mice, these are the same strain, from a different vendor. So instead of from Maine, Bar Harbor Jackson Labs, they got it from Tacoma out in Washington. When they repeated the experiments, the tumors grew, even in the presence of the immune checkpoint antibody. And they couldn't figure out what was going on. But one of the graduate students, or postdocs in the lab, said, wait a second, maybe the gut bacteria are different. Nobody would have thought of that. I wouldn't have thought of that. They then took some of the animals and they actually mixed them together. So those of you that have dogs, you know that dogs do have a bad habit, coprophagia. They'll eat stool. And in fact, for most species, and very rare ones don't, eating stool is actually an evolutionary positive thing, which we'll get to later. It's a little crazy, but probably valuable. And when they mixed the mice together, all of a sudden, the Tacoma mice were responding to immune checkpoint therapy. And they were able to identify some organs. So I said, well, if that's true in mice, I'm going to jump on this because I'm a clinician. I can be fast. You know, I called them up because I wanted to collaborate, and they were real nasty to me. You know, we're University of Chicago. We don't mess with low life in Dallas. So I said, to heck with you. So I worked very hard and put out the first report showing that there are specific bacteria in the gut that will predict response to immune checkpoint therapy. Okay, and that's Faecalibacterium prausnitzii, Bacteroides thetoidomicron, Hodemania biliformis, and this has now been confirmed in Paris and in Houston. Now, other groups have done similar work, finding similar organisms, Faecalibacterium prausnitzii. So this is the group in Paris, Ruminococcus bromii, which is related. And then here's another group finding a different bacteria. This is the group in Chicago, and theirs is probably wrong. And then here's another group in Paris that shows, interestingly, for kidney cancer and lung cancer, it's a different bacteria. It's Akkermansia muciniphila pops up. And you say, well, that's common bacteria in people that drink red wine. It's Paris. But my preliminary data when I was in Dallas showed that for those cancers, that bacteria showed up for kidney cancer. So that's getting us back to what I'm trying to do. So I've now gotten about 120 different stools from patients going immune checkpoint therapy. Any of y'all know people that have had it or that are going to get immune checkpoint therapy, please let me know. Maybe I can talk to them and get a sample of the stool. We pay for all the sequencing and analysis, and then we can maybe be able to give them a hint. If they respond, then they're just helping others. If they don't, we may be able to recommend some probiotics that'll help. Okay. Now, here's the research. This is what was my main theme with you guys. Correlate the gut microbiome with immune checkpoint response. This is what I do in the mornings from around 5:30 a.m. to around 9, is go work on the mice to reproduce some of this results. And then my collaborators analyze lymph nodes, bacteria and dendritic cells to figure out the molecular mechanism. And then we're doing bioinformatics to see if we can explain why certain bacteria work for certain tumors and other bacteria for others. So this is where we are as of a week ago. Samples with P is progression, U is unknown, R is response. Samples we've collected. I've sent off 15. Interestingly, China will do the sequencing for me at half the price of sequencing labs in the United States, which is sort of sad, but I've got to go with the lowest cost. but it still cost me about $200 per patient to get the sequencing. And part of the reason, I'm embarrassed to say, that I came to Alabama is that I was given what they call research package that would allow me to spend money on that. And you say, well, why don't you get a grant, Dr. Frankel? Well, because they want preliminary data, so you're sort of caught in this catch-22. So this attracted me that I'd be able to get this data and confirm and expand it and see if there are different bacteria for different tumors, and then be able to help my audience. In addition, I've been working with a company called Vedanta Biosciences in Cambridge, Massachusetts, and they've created the probiotic cocktails and patented the bacteria we had, so maybe it'll be useful.
How long have you been here?
A year. Got here July 10th, so no, it's a little more than a year and 2 months. So this shows what I've been doing in the laboratory. If you take those organisms, you see that if you give immunotherapy alone to wild-type mice, looks great. But if you give antibiotics, so you're cleaning out the bacteria in these Jackson mice, and you give them Lactobacillus acidophilus, this is what you get in yogurt. All right, that's not so good. It's not doing much. If you give them the specific organisms that we found help the humans with melanoma, it appears to help the mice respond with melanoma and immunotherapy. And then this is just a very over-busy slide saying the same thing. VE800 is the commercial brand name for those bacteria I just showed you doing the same thing. Okay. And there you don't have to have stool injected into you, you take capsules. Now my personal bias is the stool works better, but I'm not going to say that too loudly when I'm working with the company. It's also cheaper. It sounds ugly, but if you have cancer and you want to do better, it's not that hard a thing to do. So what do we know about how it works? So the bacteria actually do make it through those M cells. They meet the dendritic cells. They then go to the mesenteric lymph nodes, and there they tell the T cells, hey, don't die. So normal control, the PD-1 marker on the T cell says, I've had it, it's just not worth fighting anymore. I'm not going to fight cancer. When you use immune checkpoint therapy, you can lower that number. That's preventing the T cells from giving up. But if you use just general antibiotics on someone, then you see that all the T cells give up. In fact, we've been able to show, and there's now 2 publications, that if you take antibiotics in the month before or the first month of immune checkpoint therapy for cancer, your response rate goes to almost 0. So I will not let patients, and sometimes you have to if somebody has sepsis, but we try and avoid patients going on immune checkpoint therapy, getting antibiotics, because you clear out your gut bacteria and they can't help. Okay. I'm not showing you this. This just gets into, this would be good for the basic science, but because this is where you co-incubate, but it's not needed for this talk. What is interesting is that my partners in Dallas, the bioinformatics people, we looked at melanoma, neoantigens. This is when you have a cancer. Don't copy this because because I haven't published this yet, and my partners will kill me if this. But I have to have fun. So if you look at, this can be done by bioinformatics. The melanoma neoantigens can be taken, you what's called in silico, break them up into pieces, and then have them fit into the HLA components for the T cell receptor, HLA-A2 usually, and then how does that match up with antigens obtained from gut bacteria? Okay, and lo and behold, if we look for these neoepitopes that align with the gut bacteria and look for a response, then the responders, there they are. Faecalibacterium prausnitzii, Holdemania biliformis, Bacteroides theta-myota micron, a couple of others that we're studying now in the lab as well. But these are ones where you're showing evidence that the patients respond, and they have the antigens that cross-react with the tumor. So in essence, what it's saying is there's some bacteria in your gut that may work like vaccines. Now, we haven't proven this. This is called a hypothesis. But we're going to look to see if that holds for the different cancers, for kidney cancer, melanoma, lung cancer, if you can match up which bacteria with what antigens that they may display. In essence, not only be immunostimulatory, but prepare your immune system for the particular antigens. All right, so the final model, because this is what you have to do when you want to do science, you like to say you can all make it into one slide, one story, even though you spend your life and usually disprove whatever you put together. So here are the bacteria, and the gut immunostimulatory bacteria make their way through the gut wall, across the M cells. They reach the mesenteric lymph nodes, and there they turn on the dendritic cells, which then turn on T cells, which then go and attack the tumor. That's the model. So what we know so far is that there are gut bacteria that impact human immunity, behavior, and trigger multiple human diseases, that immune checkpoint therapy produces long-term remissions, but there are side effects, and that certain gut bacteria may improve the immune checkpoint therapy response, both in patients and mice, so 2 parts of Koch's postulates. And then finally, the active bacteria may stimulate dendritic cells and educate those cells in the mesenteric lymph nodes to infiltrate and kill tumors by at least 1 or 2 mechanisms. So these are the questions. You've got to have continuing questions in order to try to get grants, in order to have an excuse to do your work. And one is, nobody has proved in one laboratory that different bacteria work for different cancers. And now I want that to happen in Mobile, Alabama. Okay. Do different geographic areas use different or the same bacteria for different effects? In other words, does diet make an influence? And we're going to study that. There's 2 papers that have come out in both melanoma and kidney cancer that if you're overweight, you actually have a better chance of responding to immune checkpoint therapy. And it's sort of counterintuitive. We think it's somehow related to the gut bacteria because I told you there's a connection. We haven't proven that. What is the optimum probiotic to give and how to give it? We're trying to learn how to do that. I'm going to Rehovot, Israel, where they've been very mean. They've said all these probiotics that we're using basically don't work worth a darn, and there's pretty good evidence to support that, except in some mice models. So I want to learn from them what they think are solutions to be able, what they think is you're going to have to use fecal transplants. But I want to talk with them face to face and see how they're doing it. And then finally, learning the biology. How do they work? Is it by just stimulating the immune system generally? These are pathogen-related receptors, or are there neoantigens? Is it working like a vaccine? So again, for those of you out there that know people with cancer, then they get immunotherapy, please have them call me. We'd love to work with them to get a sample, even if they've had it a year or 2 ago or are going to get it. And then I want to thank my collaborators, Andrew Coe at UT Southwestern. He's a microbiologist who really taught me all the steps so that I could analyze the microbiome. Bruce Roberts at Vedanta has been supporting the work and providing probiotics from Massachusetts. Kenya Honda in Japan has been advising and collaborating. He's found a group of bacteria in normal volunteers that had these properties, and we're testing those. Chandrasekhar works on the dendritic cells. Chulan's my biostatistician. These people you may know. Alexander Brown's a oncologist, Pensacola. Tracy Petty is my research lab partner. Mohammed Cushman's a doctor here who's been supplying both a large number of patients and is now going to partner in and help run my trial. Amit Reddy is a postdoc working with me on this, and then Michelle Mock is my administrative assistant. And I want to thank all the patients and the referring doctors, funding sources. Thank you. Questions?
When you've looked at the gut biome and deficiencies, have you seen any correlation for the incidence of cancer as related to deficiency in gut biome?
So It's a complicated question for two reasons. One, can you use it as a predictor? Or it's complicated because you're saying we're going to measure people's gut microbiome, which costs not a trivial amount of money, because in addition to that $300 for sequencing, you may be able to get it down if you 16S ribosomal to $75. You still have to extract the DNA from the poop. You have to get an informed consent. So then you're saying we're going to wait 10 years and see if they develop cancer. I can't get a grant to do that. Now, there are people that have been looking at Fusobacterium nucleatum in colon cancer to see how predictive it is, and it's still not completely worked out. So colon cancer is the closest for that. I have not chosen that research project. It's extremely difficult. The other complication is if you have someone that's not well, that may be going to get cancer in a year or two, they're already sort of on their way, they may have dysbiosis, meaning abnormal gut bacteria, but it may be a consequence of the cancer rather than the cause. So it's not easy to dissect that. In other words, many cancers are an inflammatory state. There are scientists who say that cancer is a form of chronic inflammation. So if that's going on, it may be the cause of the abnormal gut bacteria, not the
But would it not be a predictor?
We don't know that. And as I say, a lot of science now, we're just going to have to accept is probably not going to be on the table, at least in the near term, because we're spending all our money on the military, on, I won't say making billionaires richer, but, you know, because I know I'm in Alabama, I've got to be careful. But even with some improvements in research funding, to do some studies, you just have to be careful because they cost a lot and you know the payoff. So part of the job of people that do peer reviews is to say, if I'm going to give somebody $100,000 or $1 million, what is the chance I'm going to get an answer out of it that I can then go to the next step? What you describe is a very important question, but I don't see it as an easy one to tackle outside of that Fusobacterium nucleatum and colorectal cancer. And even there it's tricky because that bacteria seems to give trouble when it's worked its way into the wall of the gut. Well, then what does that mean? That we have to do colonoscopy on every person and biopsy their gut? Well, that's not cheap. That's what they're doing in Israel where I'm going to go talk, but we can't routinely do that. So I think it's a good question, but honestly, I'm probably not going to study that, not in my lifetime. Other questions? Yes. All right, you want to go?
You hear at times about medical professions, usually through email or advertisement, about colon cleansing. What's your thinking on that?
All right, I didn't show you, but we have good data that using either antibiotics screw up your gut bacteria for about 3 to 4 weeks, and then they come back. It's the same ones as before, but you know it's not good during that period. When you do a colonoscopy prep, which is like a colon cleansing, or tape stuff like that, it has almost no effect. It reduces what's in your gut for about a week or two, and then they come back. You say, well, how could that happen, Dr. Frankel? Because number one, remember you have 100 billion bacteria in the gut. A lot of them are actually stuck to that mucin layer or embedded in it. So you get rid of all this, and the ones that are there, remember evolution has worked. We've been evolving with these bacteria for at least the 50 million years that humans or pre-humans, mammals have been around. So you know, we're a tight, you don't change by the age of 4. You don't change your bacteria. I could identify anyone in this room by typing your gut bacteria. In fact, my wife was, I shouldn't tell this story either. My wife became head of the HOA, Homeowner Association. I didn't even know what that meant until we were in Dallas and she wanted to, and the reason she wanted to do it was she's very upset because we had this little 22 house, you know, little community where people were walking their dog and not picking up after the dog. So it's just driving her nuts, and she did succeed in life. One of her claims to fame in life is we have a little, they make these little boxes with the little thing, encouraging people to use it right in the middle of our little development. But I wanted to say, hey, I can find the culprit. Because for $50, you can take some of that poop, you can sequence the dog poop, and you can go through, you do have to get a sample from the dogs in the community. I could nail that down. But the HOA wouldn't give me the money. But you basically, you have your gut microbiome, you don't change it. So although I told you about we're trying to do probiotics, we haven't figured out exactly how we can get the new bacteria to engraft. In mice, I can do it. I have to give them very strong antibiotics, and then I just keep pouring the bacteria I want in with no other bacteria. It's sort of hard to do that with humans. Humans, you saw that one study in autism, they gave a lot of antibiotics, they did do bowel cleansing, and then they gave massive amounts of the bacteria they wanted over and over and over again. And there they were able to get some engraftment, but it wasn't easy. So in general, bowel cleansing do nothing. Antibiotics may immunosuppress you for a few weeks, but your gut bacteria will come back. So you go, when you're born, you get the bacteria primarily from the mother. It's impacted by the breast milk, so you have the bifidobacterium, and then in the lactobacilli, and then by age 3 or 4, you get your adult collection, and then you've got it for the rest of your life. Now, they're not easy to change that, and certainly a bowel cleansing isn't going to do it. And that, separately, it's a little crazy anyway. Now, it is true you feel better when you don't have a lot of poop in you, but that's not really, it's not a good way to make your life. I would not recommend that. Yeah.
No, I've heard some doctors, if they give you antibiotics, they tell you to go out and take like acidophilus or some other type of
Yeah, it's baloney.
probiotics with it.
So you should avoid antibiotics unless it's life-threatening. The one thing I've learned here is, antibiotics do dramatically change for a period of time your gut microbiome, and that's going to change a lot of things. You'll notice your GI tract will often do weird things. You don't really feel the same for a period of time. It's not trivial. And we've used antibiotics far too much in our society. And even in my oncology community, I have to fight people from pouring antibiotics into people. And when you see the data, I didn't show it in my talk today, but if you look at the data of cancer patients who are going to get immune checkpoint therapy that got antibiotics during that first month before, during, the response rates are just terrible. So I would be careful of antibiotics except when you need them for life or death. And I saved my life once, but I try to be careful. I would not use bowel cleansing unless there's some physiological thing that your GI doctor has to do it. Yeah.
So, there's a lot of stuff out on the market now, probiotics.
Yeah.
I mean, is there anything that's, the different type of?
So they're probably,
It would be good?
so they're probably pretty much all just you're giving money to these companies, and that's a fine thing to do. I mean, I guess if you want to help them get rid, but I did a study. It was in my paper that I published, where I had some patients who did take probiotics. I looked at what probiotic they got. I looked at their bowel gut bacteria. It just like washed right through. There's nothing. And the reason is you have a community in your gut. And when you try to play around with putting something else in, it just says, no, thank you. And that's actually self-survival. We have evolved with our bacteria, and if you randomly put something in, it's probably not going to. It takes a lot of hard work to get a probiotic to take, and we're trying to figure that out right now. But buying them in a store doesn't even. Now, having said that, there's a little bit of data that Bifidobacterium longum and some of the lactobacillus species in autistic children do seem to help a little bit. So I don't want to be blanket about that, but that's a very special case, and the benefit is marginal, maybe 10%, 15%. Yes?
Is there any evidence that there's a relationship to celiac disease and gut bacteria?
Yes, so it turns out, after you read this first book, I won't give you the name of the second book until you read Ed Young's book. Now you can get it I make no money off of him, but it's really a fun book. This is like $11 on Kindle and $14. Again, I Contain Multitudes by Ed Young. The second book, I won't give you the name, but it's by Martin Blazer, so you probably can figure it out. But he was the one that discovered Helicobacter pylori with gastric cancer and its absence with esophageal cancer. And as he did his studies, he discovered, and others did too, epidemiologists, that people that had been brought up too cleanly get celiac disease, get peanut allergy, get asthma. In fact, the Western sophisticated world, wealthier people from about 1860 on started getting all of these allergies and autoimmune diseases because they didn't have a complex gut flora. So that would be book 2 if you get to that point. That's a separate story. If you have grandkids or kids, just put them out in the dirt, make them eat dead. And if you're lucky, they'll get some pinworms. And you think I'm joking. I'm not. You need to be exposed to these. And what that does is it makes you a robust immune system, and you won't get allergies or asthma later in life. Right? Yes.
After you.
Go ahead.
Is there any relationship between all antibiotics we feed animals? and-
Between what?
We feed, I think over half the antibiotics in this country are consumed by animals.
Yes.
So we as humans eat most of those animals.
Yes.
Does that have any
So
effect on our gut?
in this book, and in the second book, it'll show you that almost all of protein that we get, not from plants, but from animals, has been exposed to large amounts of antibiotics. And you say, Dr. Frankel, why is that? Is that to protect them from illness? No, it turns out that in animals, probably including humans, if you empty out the gut flora, that sends a signal that causes obesity or growth. So the size of the cow or the chicken will almost double in size. And I see it in the lab. Like I have a group of animals that I give antibiotics to as part of my control, right? They just grow right up. So you say, well, why would that make a difference? 20 to 40% increase in growth rate in an animal, if you're a dairy farmer or whatever it is, it's a lot of money because you're not paying anymore. The antibiotics are cheap. So they tried to make legislation to reduce it because of all the antibiotic resistance organisms that were developing. It turns out they found loopholes around it because it's just so vital to the agriculture industry in this country. It is almost impossible. Even when you get, quote, organic produce, you got to be careful because they'll say they have ways of getting around that even. So it's a real problem. I mean, on the other hand, if you're trying to make a better football player for Alabama, I suppose you can give your kids antibiotics when they're real small. They might do that in Nebraska. Yes.
Yeah, so can gut microbiota act as a biomarker to predict who will respond?
Yes. Well, that we already have. Now, you say, well, why don't we do that? We can. We can make what's called an RT-PCR. This is where you can take the bacteria in the gut, do a quick extraction of the poop, and then do a chemical reaction called the polymerase chain reaction, a little test tube with a little marker dye, put it in the machine, and within 8 hours, know whether or not that person has the organism or not. That would be a nice project for someone as a postdoc or whatever in my lab. The good thing about that is you might encourage someone if they have it. If they don't have it, I guess you're going to give them bad news. Maybe it's tricky because we don't yet really know exactly how to give the probiotics so that they'll stick. So although I tell patients I'm going to help them, I can't tell them for sure that I can tell them. So I think that's the way we're going to go. But you can see that sometimes you can give people bad news and not be able to do anything about it. For example, Huntington's chorea, if you're born with the Huntington's mutant gene, then when you're 50 years old, you're going to be like Arlo Guthrie, I guess, and your brain's going to go. And there's nothing you can do about it. Well, that may change with CRISPR-Cas in the future, but I don't think yet. So there are things, with BRCA1 and 2 for breast cancer, you can do something about it. You can do surgery, and we have new drugs. So I think eventually this is an important assay to have. And we pretty much are ready to do that now.
Is this what you're saying that the Israelis say they have it, they know how to do it to make it stick versus
Yes.
our approach?
Yes, I got sent the articles from Dallas 2 days ago. I stayed up last night or night before last, reading, very dense. It's 2 cell papers back to back, which I wish I had, but they have. And in it, they explain how we're doing things wrong and how you have to do them. So I'm actually going to, my wife has insisted we're going to go to Israel in February, so I emailed him, so I'm going to give a talk there at Rehoboth at Weissman in February, and hopefully I'll learn. I mean, I can read the article, but they're so hard to figure out what's going on that when I meet, I hope he can give me a better sense of how we can solve getting that trend. And it may be we're just going to have to do fecal transplants. It's not the end of the world. And even though you say, well, I really don't want someone else's poop in me, if it saves your life, it's to me a lot easier than getting a bone marrow transplant from someone you don't know, which they are a lovely person to donate that, but this is a little easier. Yeah. Yes.
Is there any truth that sugar causes cancer to grow?
Yes. So it turns out, and this really pains me because I had bagels last night, and I love pasta, and I love sugar. They had some treat where you give $1 or $2 and you got some treats over at the Mitchell Cancer Institute yesterday if you donated. So I got this apricot thing. It's just fabulous. So I love sugar. I love chocolate. The problem is that on several fronts. One is obviously you load your body with small sugars, because when you eat bread, you think, oh, that's not sugar. Well, unfortunately, amylase in your mouth converts it to sugar, darn it, and that's why it tastes so good. All right. So the sugar is both bad for our gut. It tends to cause the proliferation of what's called Bacteroidetes, which is a class, one of the phylum bacteria that are associated more with cancer than without, just in general terms, versus the firmicutes, which you get when you have more protein and slightly different diet. So I would, just on a basic level, there are prebiotics, and eating straight sugar is really not good for you. Sadly, a paper just came out, again on the gut microbiota, that when you take NutraSweet or Sweet and Low, which I do, it turns out that turns on organisms in your gut that metabolize that, and you end up gaining weight. I'm so upset about that. That came out again from that darn Israeli lab about a month ago, and I am really upset because I haven't figured out how to solve that. So what you can do is this Mediterranean diet really overall is much better for you. And in a variety, it reduces inflammation, probably reduces arteriosclerosis a little bit, probably reduces the metabolic syndrome a little bit, probably has some impact on cancers. In terms of established cancers, there was a guy named Milken who got prostate cancer about 25. He used to have penny bonds or whatever. He was whatever it was in the stock market. He made billions. And then he got metastatic prostate cancer. He's still alive. So he's been on this very extreme Mediterranean type diet. Now he has the benefit of being a billionaire, so he can have a full-time chef making him, wonderful meals. Most of us don't have that option, but to the degree that you can eat a Mediterranean type diet, it is better for you both in terms of inflammation and probably in terms of cancer. The data that established cancers use sugar, we know that. There's something called PET scans. Some of you are probably familiar with it. PET scans are basically a radioactive sugar, deoxyglucose, that's got a PET emitter on it, so that's why it's called PET, positron emitting tomography. But basically, you're taking sugar into the body, and those sugar molecules go to the tumor. And we explain that is that there's a Warburg effect, And that goes back, we could go back to several billion years ago for how these archaeobacterium survived. But that's another story. Just in general, if you can reduce the overt sugar intake, you're better off. Yeah. But that's not based on really anything I'm showing you here today. Yes?
There are conditions like bronchiectasis where the preferred treatment is a regimen of very strong antibiotics. Are the side effects to something like that worse than the condition itself?
No, I mean, I want to be careful here. Antibiotics do have a role. I don't think you want to abuse them. But inflammation in your major bronchial airways, you need to cut that down. And unfortunately, it's not part of my talk today, there's a different set of bacteria that love the airways and that produce something called a biofilm. Whole different story, different talk. Pseudomonas is a big part of that. And so you have, so your pulmonary doctor is going to do everything he can to reduce the biofilm and inflammation, and so they use antibiotics for that. And you need a lung to survive. So I don't think you have a choice on that. It's same with children with cystic fibrosis. They've got abnormalities in their mucin. They're having to do things to try and survive. Yeah. Yes?
Any benefit of yogurt with live culture?
So, the lactobacilli may have a small benefit in if you're a really angry person. It may have a little bit of a calming influence because that's part of its goal for the infants, but you're not going to get much of it. There's a lot of calories with that. So I did something, there was a drink that has all this stuff in it, kombucha. So I was in Dallas and somebody said, oh, you need to do this. And so I started drinking it, and I didn't really love the taste, but I thought, maybe it's good for me. God only, we don't know all the answers. But I started gaining weight on it. So, 'cause if you read the label carefully, there's a lot of calories in with that stuff. So I think you've got to be careful. As painful as it is, calories are calories. So when you choose foods, you want them both to taste good, if you can, but not, unless you already have cancer, and then you need to be overweight so that the immune therapy works better. But in general, I wouldn't recommend that. So I would say, if you like yogurt, eat it, but otherwise, there's nothing special about it.
What about fermentation, like fermented foods?
Right, well that's what the
I mean, it's
kombucha
not,
is.
not something that you buy, something that you can make, for example, like sauerkraut, something like that, like homemade,
Right, well now sauerkraut is probably a good vegetable. And I want to be careful. People that give you nutrition talks and they think they have all the answers, you need to be very careful. We don't know hardly anything. So I've been talking to you about bacteria, which would be at the probiotic or later stages. And as I just told you, in Israel they're saying I'm looking at the wrong bacteria because it's the ones that are right on that mucus lining that are important that I can't get to in the stool. But now if you tell me what's the relationship between the nutrients and the bacteria, I think it's very complex. And I don't think we know very well. So I am testing yeast in my next experiment to see if, because it popped out as one of the organisms when I showed you in melanoma that if you have yeast in your gut, it may help. So we're going to test that. But that's, we don't have the data yet on that. I would say in general, eat what you enjoy, but don't eat something because you think it's going to cure all your diseases, with a possible exception of olive oil. I don't think you can get too much olive oil. Yeah. Yes.
I'm back to the sugar. What about natural things like plain sweet potato, bananas, fruit?
So that's the very sad thing. I eat fruit because what my wife says is if I eat apples to get the same amount of calories as a little bag of cookies, I'd have to eat a ton of apples. But it is sugar. So we tend to eat in my home grapes and melons, things where, because I love food, things that limit how much sugar I'm getting per unit time. So one of the things you should probably, not a great thing, is a lot of people think they want to juice things, convert all this stuff into this liquid and our soups. Those things are just loaded with calories, and I don't think you're not enjoying it as much. It's personal bias, but you can do whatever you like. I would just,
They push baked sweet potatoes and that was
well, I mean, the amount of calories you get in a baked sweet potato versus a bowl of cereal or a cake is probably less. So, yeah. So I mean, you have to use judgment. It's very hard. We're humans, and in fact, we've evolved probably to like a lot of carbohydrate. In fact, I think there's been some shifts in population that occurred probably 10 or 20,000 years ago as we became somewhat agricultural, that people have gotten sweet tooth. And it's very hard to just say, oh, we're not going to do that. Yeah. Other questions?
So it's kind of food is medicine.
Huh?
It's kind of food is medicine.
It is true. So you didn't see, I jumped over it, was that they looked at the gut biome in people in tribes in Venezuela and in Africa versus people eating McDonald's in the United States. And we don't know yet what it means, but in Africa and in South America, the more natural diet consuming people had 300 or 400 species, 3 or 4 phyla in their gut, never got allergies, never got autoimmune disease. And we eat our cheeseburgers and hamburgers or whatever, processed food. That's probably not good. It's certainly changing our gut microbiome. But exactly how, I don't know. That's book number 2. You read the first one, the second book by Martin Blaser goes into that, but it doesn't offer a simple solution other than, having a varied diet. Yeah. Other questions? Yes.
So does the microbacteria in your gut change if you eat a different diet
Yes, a little bit.
over time?
A little bit. So I told you that I could tag you or identify you, and the answer to that is yes, because you have an average of almost 200 different species in your gut, and some may change a little, almost like a football team or something. Others will, I could still identify you, but you can modify it slightly. To ask where a major change would take many years, and nobody's done studies they've successfully done that. They've tried. The Knight's group at Scripps and UCSD in San Diego, they tried prolonged diet changes, and they did it by, what do you call it, cloud funding, which I have, I'm not sure we're supposed to do that at a university, but they were able to do it there where they got people to both go on a diet, send them poop, and cloud fund their work. And that's how they're doing it. So they have these gorgeous sequencing, because $300 per person is cheaper than it was, but it's still not a trivial amount of money. They've cloud funded it, so they have their own, there's a more advanced way to sequence DNA, where a single strand of DNA goes through a little hole, and they've got that working in a sort of a factory setup for people there. And they publish good work. They have very good bioinformatics, but they got a lot of their funding by something called crowdsourcing. Yeah, I'm not sure we're supposed to do that. But relative to your question, we're not at the point where we can connect what you eat with your gut microbiome with outcome. If you read my paper, I had a statement in there that tree nuts were associated with a higher response, and I found a chemical that seemed to be connected to it. But when I tested it in the mice, it didn't work, so I don't talk about it. So things come up. It's called beta era. Things come up when you look at 40,000 pieces of data. You say, oh, people that wear red shirts are terrorists. Well, where'd that come? Well, maybe, that day, looking at 200 people, that association, but it's not necessarily true the next day or in a larger set. And that's what can happen when you have too many numbers, is that you can get misleading stuff. So I think, for me, unless I was able to prove, Koch's postulate was they found anthrax in people that were dying of anthrax. They took the organism, they put it in a mouse, killed the mouse, also killed birds. Okay. So that was what's called step 2. Step 3 would be taking the anthrax and putting it back into someone, but they didn't do that. So the third part of Koch's postulate, they used a vaccine or an antibody to anthrax to reverse the disease. Okay, so that was their substitute for putting it back in a human. And that then fulfilled the 3 codes. So we're trying to do that. 1, is there bacteria that predicts response of immune checkpoint therapy for a given cancer? Yes. 2, does it work in the mouse? Yes. When we take a clean mouse, put it in, they respond. 3, if we now take that bacteria and figure out a way to get it to stably stay in humans, will it improve the response rate in humans? And we hope the answer to that is yes. And we will be the phase 1 site for that in the United States with this company. But as we talked about, we haven't figured out completely how to empty out the bacteria that are there to get the new one in. I think we're going to have to do fecal transplant, which interestingly is very cheap. They're not going to make money because you don't have to have a company to do it. So what De Palma is doing for his C. diff patients is he's just having them do it at home with one of the family members. And they follow his instructions and then they're cured. So I don't know if we're going to get to that point, but that's where the best research is looking. And in Israel, in Tel Aviv, they have a separate hospital where that's what they're doing. They're taking people that have recovered from immune checkpoint therapy, they're taking their stool, they're giving it to people that are about to go under immune checkpoint therapy. So.
How often would you have to repeat the fecal infusion?
Well, it turns out once you turn on T cells to attack the tumor, it may work the rest of your life. It's almost like once you allow your immune system to recognize that you've got a cancer in you, oftentimes that'll hold you, as far as we know, for the rest of your life.
Would that help for weight control?
That we don't know yet. I mean, the early data looked encouraging in mice and in some patients, but that's very complicated, and you run risk there. When you put someone's stool into you, maybe they will or won't lose weight, but maybe then they'll get autistic, you know. It's not, I think you have to be careful. If it's a life-threatening thing, yeah.
It would be a gold mine if you figured that out.
Well, I don't know if people would do that. I don't know. I don't even want to think about that. All right, thanks, all guys.